blinding and blinding issues, because it's a big problem. And so I'm really looking forward to the two talks we're having. Dr. Jean Mulinde, again, who had been previously introduced, and Gail from MHRA. And we also have one of our third and final speaker is Dr. Julia Cho. Now, she works for the FDA. She is currently director of our Division of Generic Drug Bioequivalence Evaluation in our Office of Study Integrity and Surveillance in our Center for Drug Evaluation and Research. And her division provides an oversight to bioequivalence bioavailability studies submitted to FDA in support of both new and generic drug applications throughout on-site inspections and compliance evaluations. So Julia joined FDA as a review in the Office of Pharmacology and was a reviewer and team leader in the Office of Scientific Investigations in the Office of Compliance, where I had originally met her. So I'm looking quite forward to our next session. So Gail Francis, I think it's our first, or Jean's our first speaker.
We're actually tag teaming.
Oh, together. Yeah. That's why you're confusing me. Okay. Anyway. Okay, so please turn your attention to them. Thank you very much.
So as I said, Gail and I are actually tag teaming this talk, and we'll just be going back and forth, and we're going to do it fireside chat mode here, sitting here at the table to talk with you. So the title of our slide, Unblinding, Let Me Count the Ways. That probably gives you some idea of what you're going to be hearing about as we move along. So first, the learning objectives. So basically, that you gain an understanding of the basics of data, how our data flows through the various systems, if you don't already have those basics, how it's collected, integrated, transformed, analyzed, and reported in submissions. And that you understand there are many, many ways in which clinical trial data may be unintentionally unblinded, and that premature unblinding of data, that being before database lock, that permits identification of subjects' treatment allocation, may really raise quite significant concerns related to the adequacy of data integrity and data quality in your clinical trials. So we're going to start out with this kind of very complicated looking diagram, which in fact really isn't complicated when you think about it. But we did think it captured the high points of how data is flowing through the data life cycle and the data flow. And so importantly, one of the things I wanted to point out to you is that for every arrow you actually see on this diagram, we've seen cases of unblinding. So the many, many pathways of unblinding again. So Dr. Temple this morning talked about why we blind, and I'm just going to remind us of a few things. So we blind studies to prevent sites, patients, study teams from being aware of the treatment assignment, to prevent the introduction of intended or unintended bias into how the study is being managed and the patients being managed in the study. And even in the case of an unblinded study design, with the exception of data being reviewed by the Independent Data Monitoring Committee, we generally also mask treatment assignments in data sets to prevent any performance of aggregate data analysis by treatment arms. Again, to prevent shifts in the management, either intended or unintended, in the clinical study. Importantly, even though we think, in particular, trials with subjective endpoints have a need for blinding, really even in trials with more objective endpoints, there's a risk that individual subject management may be altered when people know the study treatment allocation. Thus, there's really a need to very robustly maintain your study blinding procedures to protect the integrity of your data. So again, Dr. Temple this morning touched on some of these considerations. This is not by any means an extensive and complete list of considerations for you to keep in mind, but we just wanted to point to some in particular. So things that can unblind your study. Obviously, the appearance of the investigational product and how well it's matched to your control or placebo can be a source of unblinding. Then there's those obvious and known side effects for particular products that may result in unblinding through that pathway. Likewise, effects that are well known on laboratory parameters may result in an unblind situation. And then we have many reports that we're using to manage our studies and operationalize our studies. Adverse event reports, assorted IRT reports coming through, the many, many different internal reports, all of which we need to maintain study blind on as appropriate. Then we have things like batch numbers, lot numbers, kit numbering, expiry dates that are applied to our kits, and they are sometimes also a source of unblinding. We touched on, again in this morning, the importance of data masking procedures in our data flow, and the all-important control of user access and user privileges in our various systems. So with that basic beginning point, we're going to go back to our diagram and we're going to start talking through some examples of some things that we've seen. So we're going to start here with the labeling of investigational products. So examples related to labeling that we've seen include numbering patterns that actually permit subjects, site staff, and study teams to guess the treatment groups that they're in. And this has to do with batch numbers and lot numbers sometimes, expiry dates, or the manner in which kits are actually numbered. So I'm going to walk you through an example of sequential kit numbering and how that might unblind your study. So this is basically the picture for one site that's involved in your study. And the randomization code for the study is on the left, and of course the site can't see that information. The site's initially stocked with kit numbers 2001 through 2004. So they enroll their first subject, subject 101, who gets randomized to active product. This subject gets kit 2001 assigned to them. They enroll the second subject, who is also randomized to active product. And this kit assignment is 2003. So your site staff immediately see that there's been a skip in the kit numbering. They may begin to become suspicious that these two subjects are on, and in fact, the same treatment. The other thing that happens here is now we have only the green kits, which are our placebo kits, left at the site. So the site's going to be restocked with another set of kits. So they now get kits 2005 through 2008. It doesn't have to be those numbers. It's just important that they're sequential numbers here in the example. So at this point, our subject 101 comes back for another visit and another drug we supply. And so what happens is they receive kit number 2007. So now your site is really thinking, there's two kits left in my original shipment. This one's pulled out of my next shipment. These two subjects are probably on the same treatment arm. Subject 103 is enrolled, gets randomized to placebo, and receives one of those kits from that initial shipment. So they get kit 2002. So now your site again is thinking, I know that my first two subjects are on one arm. My next subject is probably on the other arm. Subject 104 is enrolled, randomized to placebo, and gets kit 2004. So they may not know specifically which treatment arm the subjects are on, but they think they know that they're on two different arms. And they may make assumptions that they do know what treatment their subjects are on, which of course may introduce biases in the way they manage the subjects. So with that said, I'm going to pass off to Gail.
Thank you. So obviously the way that the IMP looks can also unblind. So for those that are administering it, those that are monitoring it, and obviously the patients that are taking it as well, especially if they talk to each other and compare notes, which we know from practice that that's exactly what they do. We've seen numerous examples on inspection where little thought has been given to the appearance of the IMP and how that's going to be masked in terms of IMPs and placebos. We do understand sometimes it can be quite complex to try and mask the IMPs, or sometimes really expensive if you've got to then produce an extra set of matching placebos. So if the expense and the practicalities are prohibitive, then what we expect is that some consideration is given to that so you can mitigate those risks of unnecessary unblinding. So in this example that you can see on the slide, the active and placebo were very different in appearance. And as it was a crossover, the patients received both, so could easily tell the difference between the two sets of treatment that they had. And this was a particular issue in this example, as the endpoint was a patient-completed outcome. And the label can also potentially unblind, and we've seen examples where the placebo and the active have had different expiry dates. Therefore, the packaging can then distinguish between the two types of treatment. Further information, for example, to be found in the trial master file, such as certificates of analysis that had the expiry dates on, can then be linked back to active or placebo. And so with just a little bit of digging, you can actually reveal what each patient had been given.
Thanks, Gail. So now we're going to go back to our diagram, and we're going to focus on that IP shipping procedure and what's going on at the site. So examples of IMP shipping issues that we've seen, again, Gail's alluded to some of these already. We've seen cases where documentation that's included in the shipments to the sites includes kit numbers by lot and batch numbers with the actual expiry dates. And then what the site can do, they may possibly derive which subjects are on the different treatment groups based on those certificates of analysis that are in the site files. We've also seen cases where shipments containing unblinded IMP documentation that are supposed to be going to unblinded pharmacy staff wind up with the blinded staff, and so they become unblinded to what those kits are. Here's another example that we've actually seen on multiple occasions in various formats. In this case, the CRAs and study nurses at a site became responsible for preparing the unblinded IMP. But they were also responsible for performing blinded study assessments. So they're performing their assessments in really what amounts to an unblinded manner. We've also seen cases where the clinical investigator site file contains some method of randomization code or treatment cards. So in this example, we have treatment cards that were found actually stapled to the subject's medical records, thus including the unblinding material on the medical records, and then finding those medical records in the room with the clinical investigator who is making the primary endpoint assessment, skin assessments in this case. So back to Gail to talk more about IRT issues.
Thank you. So IRT, Interactive Response Technology Issues. So IRT or IVRS, IXRS, IWRS, whatever you'd like to call it. These systems have provided many examples of unintentional unblinding, and certainly in the UK we've had lots of examples of serious breaches that relate to unblinding using an IRT system. So the IRT system is interrelated, obviously, with the IMP manufacturing and packaging. It's also interrelated with the site by way of how they would manage ordering and maintaining stock at the site. It's also interrelated with sponsors, so it can generate certain reports that can then be available to sponsors, to CROs, to monitors, to data managers, to the site, depending on how those reports are set up and who's being given access to the system. So in terms of mitigating any risks of unblinding using an IRT system, this is something that needs to be understood in terms of how it's interrelated, and it also needs to be properly controlled. So this can be done via the system design specification, paying particular attention to things like audit trails, to access rights, and the design of any reports that that system might generate. So here's an example of how reports generated from the IRT system, if they're not correctly designed and set up, can unblind. So in this example, the system provided two reports, one that was labeled blinded and one that was unblinded. So access was correctly restricted to those who needed to see that information. But the issue here is that the reports, whether they were blinded or unblinded, still contain the lot numbers. So this, in fact, unblinded the treatment allocation. And unfortunately, in this example, it wasn't picked up till the 8th cohort, which was right at the end of the trial. And a review of the audit trail showed that at least 3 study managers had reviewed the unblinded reports, so it had been an issue. Here's another example of how reports from the IRT can unblind, and in this case the report detailed the number of kits that were allocated which if accessed by the sponsor, again, would unblind, because one arm required 2 kits of one medication to be dispensed, and the other arm just required 1. So access to these reports was then granted for the site, the sponsor, and the CRO, so everybody could access these. This was harder to assess in terms of impact, because the audit trail didn't go into sufficient level of detail. So these reports could be accessed from the inventory report, but to get to the inventory report, you had to go to a consignment report. So the audit trail would show who accessed the consignment report, and there were 47 users who had done that. Then we couldn't further see who'd then gone on from the consignment reports to access the inventory reports. So that happens a little bit late in the process. One of the questions we always ask when we're looking at issues in relation to IRT is whether they're at the system level or study-specific build level. If they're at the study-specific build level, often they just impact that particular trial because it's been designed specifically for that. But sometimes we have issues that have gone, that are picked up at the system level and therefore can affect more than one trial and more than one sponsor. And a lot of the serious breaches that we have reported to us go across more than one trial and more than one sponsor, so it can be quite a wide-reaching issue. Here's an example of how the design of an IRT system, coupled with the way that the trial was actually managed, can also unblind. So this is a fairly straightforward trial, so it's just active versus placebo. But each subject was individually unblinded by the research nurse at the end of their participation, and that was because they needed further management in terms of their clinical care. So an IRT system was set up to manage this, and unfortunately, or rather fortunately, recruitment was a lot quicker than expected. But IMP began to run out at the site, and a process was then put in place to transfer IMP between sites. Unfortunately, that management of site-to-site transfer was really, really poorly managed. And what that meant was there were delivery errors, and the wrong kits turned up at the wrong site, so they couldn't be used. So what that meant was, when the research nurse went to resupply the kits or randomise a new patient, the research nurse knew that there was IMP in the pharmacy, but the IRT system was telling them that it couldn't be used. It was essentially quarantined. So where the research nurse is then made aware that all the current packs in pharmacy are of the opposite allocation to the participant that they're trying to randomise, this then risks the potential unblinding when that research nurse unblinds that participant at the end of their participation in that trial. So the IRT system was a little bit at fault, but also the way that the trial was managed, so that also potentially unblinded. Back to you, Jean.
And I just would mention that we've seen a recent example very similar to the one Gail just talked about, and they actually lost an entire cohort of subjects because of those types of issues with the IRT system and drug supply. So it happens, and it's not that uncommon since we've seen it also. So the next case we want to talk about, and this is basically a finding from a sponsor inspection, where we're having a closer look at the IRT system, and in particular, we touched on how the user rights were granted for the system. So this was a double-blind, randomized, large cardiac outcome study of IMP versus placebo. It was a vendor-provided IRT system, and appropriately, it permitted unblinding in emergency cases by the clinical investigator via an IRT interaction. What it also permitted, unfortunately, was global unblinding access to over 100 clinical investigators and multiple CRO staff study team members who were integrally involved with how the study was being run. So inappropriate access, clearly.
So sometimes it is necessary to unblind, so you might need to do that in an emergency situation. And we've also seen examples where the opposite happens, where it's not been possible to unblind when you're in fact trying to. So this example you can see on the slide comes down to process. So do you remember back in the days of paper envelopes, where the unblinding envelopes were present at the site, so they were usually delivered with the IMP. The monitor would check that they were there, so you had access to them if you needed to unblind. Now this is done via IRT. We very rarely see that monitors or anybody checks that the investigators have access to the IRT system and they are in fact able to unblind in an emergency if they need to. So in this example, the IRT system wasn't working and there was a backup process in place, which also unfortunately didn't work because the investigators didn't have enough information to be able to unblind. So what they had to do is phone into a call centre to unblind. And unblinding relied on a system-related generated randomization number, which the site didn't have access to. So the site had patient IDs, but weren't given the randomization IDs, which is what the call centre needed to actually unblind. So unfortunately, that process didn't work, and there was a significant delay in trying to unblind those patients. This delay comes from the design of the system and the lack of testing of that. So this unblinding service was provided by the sponsor's help desk, who, following a request to unblind, then generated an automated email. Unfortunately, that email was designed to provide the code break in the format of another sequenced number, and that email came back to the investigator. That sequence number meant absolutely nothing to the investigator and still didn't allow them to unblind. So the unblinding in this case had to be done manually, and it took quite a significant amount of time. So this was something that was obviously never tested in part of the system setup and never formed part of the user acceptance testing. So it wasn't picked up until there was an emergency, and then there was a significant delay.
Okay, so now we're going to shift gears yet again, and we're going to talk about risks to unblinding that may occur along this pathway of samples, tissues, ePro information, even parts of adjudication packages that are being sent from sites outside of the ECRF EDC system via alternate pathways. So in this example, we have inspection findings related to adjudication packages. So the study that we were looking at was an unblinded randomized oncology study, and it was a blinded adjudication committee that was being used for the primary endpoint assessments. One site, actually multiple sites, were sending their source documentation, which unfortunately included actual treatments received documented on some of the source items. There was a vendor responsible for actually assembling these adjudication packages with the source documents from the site, along with some of the ECRF data to supply to the adjudication committee, but they also failed to recognize that there was unblinded information in the packets. So of course, this kind of wreaked havoc on how the study was seen from a data integrity standpoint, and actually forced a re-adjudication process to occur. So next up, we have that pathway of the data flow from the ECRF to the EDC and whatever sort of data repository you might be using through your statistical server process, and then on to reporting to the agencies. And Gail's going to talk more about this.
Go back one. You have to go back. Okay. So the way in which data is collected and presented in the CRA can also present risks to unblinding. So in this trial, there were two arms, standard of care arm and an imaging and ultrasound arm. The assessment was the ultrasound. So the clinical team were unblinded, but the sonographers who were performing the assessment of the ultrasound had to remain blinded to the randomization allocation because they were doing the scoring of the assessment. So what the sponsor did was to design a database and an electronic ECRF and divide it into sections. So they had an imaging part of the ECRF, a randomization part, and then also the clinical part that the clinical team would be entering data into. Unfortunately, during the site audit, it was noticed that the ultrasound pages of the ECRF also disclosed the allocation arm of the patient. So what was supposed to happen was that access was restricted for the sonographers to just their part of the CRF, and they wouldn't be able to see the other two parts of the randomization and the clinical part. Unfortunately, the design of the CRF meant that the headers stayed in the CRF regardless of what section that was, and each of those headers actually revealed what treatment arm those patients were on. So any of the sonographers that are entering the data into the ECRF could automatically see in the header which arm those patients were on.
Okay, so now I'm going to talk about findings from another inspection in which actually multiple unblinding events occurred due to systemic issues with data management practices. The study itself was again an unblinded randomized study of IMP versus an improved comparator. One product was orally dosed, the other dosed IV. And per the protocol, there are different procedures occurring in the different treatment arms. So in the IMP arm, there were PK assessments occurring and there were pharmacogenomic assessments occurring. The data management plan very appropriately called for masking of data prior transfer of data to the stats group for statistical analysis and statistical programming, really to prevent any unblinded aggregate analyses from being performed. So when we reviewed study-specific folders on the statistical server, we found a number of things. We found in the SDTM domains and in some raw data files that the actual treatment arm hadn't been appropriately masked. We found the dosing amounts and the frequencies of dosing also unmasked. And because of the IMP versus the comparator being used, that essentially also unmasked what treatment subjects were receiving. We also found in the disposition data sets the dates that PK and PG consents were obtained. And again, because these assessments were being done in only one treatment arm, you then knew who was in that treatment arm. Similarly, in the PK results data sets, the subject numbers were present, although the results weren't present, but because they were only doing PK in one arm of the study, you automatically then knew that those were the subjects on that arm of the study. Now, unfortunately, when we also reviewed the data management folder access to the raw unblinded data sets, we actually found statistical team members that had administrator privileges in these folders. So clearly inappropriate access to the data management folders with the unblinded data. What is really unfortunate about this case is that their file structure had no audit trails. So neither they nor we could really tell what access may or may not have occurred to those folders. So I want to remind you, as you're performing your study-specific risk assessment, to carefully consider all data variables that may cause unblinding by treatment assignment when not appropriately masked. As the prior example demonstrated, there's a need to look well beyond simply your treatment assignment codes. You need to look also at things like dosages and frequency, which might be impacted, routes of administration, which might unblind. And then there's things like known laboratory abnormalities associated with one arm versus the other arm that you need to consider, as are specific AEs that you might need to consider. There's also those therapeutic drug level monitoring levels that sometimes occur that need to be masked to prevent unblinding. So really, I just want to emphasize that also any variables that might be associated with one treatment arm, treatment in one treatment arm versus another treatment arm, really need to be considered for masking. And the list can be quite extensive. So back to Gail.
Okay, so coming back to our data flow then, there are also other data sources that are imported into systems used to manage the trial. So things like importation into the database, so lab reports, you could have imaging reports, electronic patient diaries, electronic patient outcomes, clinical assessments, adjudication committees. The list is fairly endless. And these can also pose a risk to unblinding as well if they're not correctly managed. Data also comes in via other sources, so via email and via portals that have been set up to manage that data as well. So in this example, the pharmacokinetic data was provided by the contract lab directly to the sponsor via email. Often for PK analysis, the lab will be unblinded, generally to save on needlessly analysing samples from those samples that you know are placebo subjects. And the lab will generally mask those results when they come back so that you can't tell which has been analysed. But unfortunately, in this example that we saw, the lab included sample analysis dates. So it was quite easy to tell from the PK report. So only those samples that came back with a date next to the analysis were obviously on the active, and those that had a blank were obviously the placebo. So it quite quickly unblinded the data. What was disappointing about this was when we had a look at it, we realised that actually this is something that had occurred previously. So there had been a CAPA put in place, which included study data transfer specification, but the lab just hadn't followed it. It affected a large number of subjects, so it affected 48 subjects. And looking again at the root cause, this had happened with at least 3 other sponsors previously, so this was quite disappointing to see. Safety reports, in particular things like SUSARs that have to be unblinded for onward regulatory reporting, can be an area of risk for inadvertent unblinding, as can unblinded reports, for example, that are sent to data monitoring committees. In this case that's on this slide, you can see the 2 different line listings were provided. So one was blinded and one was unblinded. Unfortunately, the unblinded versions were accidentally posted to the portal. And this meant that they were then widely available to everybody involved in the trial. These safety reports were at the IMP level, so actually covered a number of trials and a large number of patients. In this example, there was a really good audit trail in place, so it was quite useful, so we could determine who had accessed and opened the file, so the impact assessment of that was quite easy to determine. A lot of sponsors and organisations use a clinical trial management system. So this is something that can manage a trial's progress, so you can track recruitment, approvals, progress reports, etc. It can also manage the monitoring, and it can manage monitoring reports and generation and review of those monitoring reports. It can manage queries, communications across the team, and it can also form part of the trial master file, or at least generate reports that do reside in the trial master file. So because of this, there's also a risk of unblinding happening via this route as well. So the CTMS can have controlled access to different areas. So for example, if you've got an unblinded monitor generating monitoring reports, these obviously need to be kept separate and kept away from the blinded area. What we've seen on inspection is that we've seen issues with passwords for these systems. So sharing of passwords or not changing passwords when somebody leaves or changes a role. We've also seen passwords that are really generic, so it's just been the trial ID code, which if that's what you're doing in an organisation, it's so easy for anybody to guess what that password is going to be. So obviously this poses some problems in terms of access to those restricted areas as well. We've seen a number of issues here with CTMS, and we think it's probably because they're seen more as a sort of operational communication tools, rather than something that can really have an impact on data integrity. And of course, some general filing of documents has led to unblinding issues. So we've seen emails, data monitoring committee inputs and outputs, file notes, etc., that have inadvertently unblinded because they've been available for everyone to have access to in the TMS. And then, unfortunately, we've seen some really obvious ones. So we've seen unblinded drug accountability records being available in the trial master file, again, so everyone can have a look at them. And also we've seen CRFs that contain the randomization envelopes, which have the codes in them, but they weren't sealed. So these are just sat in the CRFs, and because they're paper, there'd be no audit trail to see if anyone actually accessed those. They were just open, they weren't sealed. So it's difficult, again, to determine what the impact of that was.
So we're just going to visit our diagram one last time here, and just remind you and point out that there's also a risk of premature unblinding of treatment assignment when data is being shared between PV systems and your central data repositories. In particular, this occurs, what we've seen, when that safety data reconciliation activities are underway. So now I'm going to turn it back over to Gail, actually, again, and she's going to talk a little bit about some related CAPAs in this area.
So when we've looked at CAPAs, what we've seen are a number of repeated errors. So this always tells us that the root cause obviously wasn't identified correctly, or that preventative actions just weren't effective. So in order to illustrate that, this is a trial that we looked at where we had some real issues in determining who got what when. So I'm sure you'll agree that's pretty crucial in any clinical trial. But for this clinical trial, there were no accountability records that were kept. So what happened for this trial, they used a set of randomization envelopes. So every time a new patient was recruited, the randomization envelope was opened by the investigator. They made up the IMP, resealed the envelope, and signed it, and dosed the patient. This created a number of difficulties because it made monitoring difficult, because you obviously needed to keep reopening and resealing. But it also didn't provide for a good, robust unblinding procedure in an emergency if you needed it. And also, don't forget, we don't have an accountability record, so it's very difficult to piece those two together. So, what the sponsor did was put together a CAPA. So obviously they instigated accountability records, and they also instigated some monthly ward rounds by the pharmacy just to make sure there was some kind of control on that. An unblind randomization list was produced. So this could be held securely in the new pharmacy file on the ward. So then the team that were drawing up the IMP would be able to go into the pharmacy and just look at the list rather than opening a lot of envelopes. So this looked like this would obviously sort out the issue and make things a lot simpler. Unfortunately, this was not the case, and we received a serious breach on the trial, because what was happening is that the randomization list was being kept in the pharmacy file, but that pharmacy file was then kept on the ward. So everybody had access to that. So this was also accessed by the chief investigator, who should have been blinded. So every time that he allocated treatment for the patient, he accessed the entire list just to see the one patient that he was going to dose. Also looking into this, we found that there were staff shortages, which is why lots of the investigators were also dosing the patients. But this led to the investigators who should have been blinded accessing the blind break envelopes again to make up the IMP. So this still wasn't working in terms of their CAPA that they put in place. So we looked at this trial again. We conducted a second MHRA inspection and actually found that it was a lot worse than we initially thought. So before there was drug accountability records in place, as I mentioned, they were using randomization envelopes, which is the only way we could tie back what the patients were actually given. They should have been stored in the CRF. In actual fact, for the majority of the CRFs, this wasn't the case, and those envelopes had been destroyed. So without accountability records and without those envelopes, we really couldn't tell who had got what and when. We also found out that the randomization list had been created by the statistician, but then given to the chief investigator, who then generated the randomization envelope. So it's actually the chief investigator who had the initial randomization list, then wrote out on individual bits of paper and then put them into the envelopes and put them into the CRF. So the chief investigator actually was unblinded right from the very start, which we didn't know it until we did the second inspection. Also unusually, when we looked at the CRFs, they included the randomization allocation. So for lots of these CRFs, somebody had written in pen in the right-hand corner of the paper CRF, A, B, C, or D, again, unblinding whatever the subjects were on. I think they'd done that just to make things a little easier for them, but hadn't really realized that anyone could access those paper CRFs and obviously quite clearly see what the allocation was. So obviously, that's a CAPA that didn't work at all. Training is something that's always included in corrective and preventative action, and that might be either retraining on an established procedure or training in a new procedure. This example shows when training has gone just a little bit too far. So at this inspection, we looked at a trial where the protocol required that half the subjects received eye drops alone and the other half received eye drops plus lignocaine. The team were trained to say that it's really important that the blind is maintained. And what we actually found was that the medical notes contained an entry on the date of the IMP administration. But unfortunately, all the medical notes said that all the patients, and there were 131 of them, had all had eye drops and lignocaine. And the reason that the investigator had written this was that they'd had it trained into them that they couldn't actually write down what the patients had had because that would unblind. But unfortunately, what it meant for us is that all we had was the medical notes that said that these patients had received both of those treatments. The accountability logs didn't help because they weren't done on an individual subject basis. They were done sort of in a bulk basis. So if there were 10 patients being dosed on the same day, we could just see that 10 lots of drug were moved from the pharmacy to the ward. So there was nothing really that told us that these patients had what they should have had according to the randomization schedule. So essentially, it meant that the data for that trial was invalidated. And that was really down to some incorrect training and a misunderstanding of the investigator. So I wanted to let you know that it's not all bad news. Obviously, Jean and I have talked about a number of ways in which the blind can be broken and obviously bias introduced into a trial inadvertently. And just like an inspection report, what we do is we tell you all the things that you're doing wrong and tell you none of the things that you're doing right and that we're actually pleased about. So what we wanted to do was also end on a bit more of a positive note. So we recently saw an example, and this is in a phase 1 unit, where a lot of thought had gone into the process of making up and administering the IMP whilst maintaining the blind. So any information that could unblind in the unit was kept in this unblinded box that you can see in the picture. And this was heavily restricted to unblinded team members only. So it was restricted by a code and also where it was stored in the pharmacy. So it was restricted by a code, kept in a locked cupboard, and the key to that cupboard, again, stored in another coded key box. So it really was fairly tight security. And then the code was changed every 6 months. What they also thought about is what people might see if they walked into the pharmacy. So in this trial, the IMP was kept in the fridge, it was in an ampule, and the placebo was essentially a saline injection. So if you looked at the stock levels of the IMP in the fridge, and you knew a patient was being dosed, and you came back later, and those stock levels were either exactly the same, or they were one less, you had a pretty good idea of whether they'd been given the IMP or given the placebo. So what the unit did was to keep all the IMP in an opaque bag, which again you can see on the picture, which had a lock on it, and the key to that lock was again kept in the unblinded box. So nobody could see whether stock levels had gone down or not, so it couldn't be unblinded inadvertently. And they also gave the same amount of thought to the returns. So obviously if you're going to just pop returns on the shelf that anyone can see, that again could unblind if you knew how many subjects were in the unit, or if you just had one subject that had come in to be dosed. So again, the returns were all placed in the locked box, and the key again stored in the unblinded box and restricted that way. So that was a really good example of getting it right, and something that we were really pleased to see, and thankful to the phase 1 unit that went and took pictures for us so that we could share that with you as well.
So we titled this talk, Unblinding, Let Me Count the Ways. And I think we've provided you with about 22 examples of how things have gone amiss in unblinding at either a site level or a systemic level in studies. But in fact, many more examples exist that have had a significant impact on regulators' confidence in your data, and that in some cases have led to rejection of data, unfortunately. We hope that we've convinced you today of how important it is for you to really understand your own data flow for your study and include this in your risk assessment planning so that you can develop the appropriate risk mitigation and risk control strategies for your data flow. Gail's also talked to you about some good CAPA, bad CAPA procedures and planning, and we hope that you have some takeaways from her examples also. So we also want to emphasize that inspectors, whether FDA or MHRA, may and will request a list of unblinding incidents on inspections. And that we realize in this day and age, such events are still actually quite common and frequently identified during inspections. And in fact, if you have had no reports at all to report, you're probably going to get a raised eyebrow at that statement. So we would like to emphasize to you that when you have had such events, the importance of being very transparent with the regulators about such issues when they're identified, so that it permits us the time to really evaluate and consider the impact on your submissions, as this may, at the end of the day, really reduce delays in your approval processes. So now on to the challenge questions. Okay, true or false? Premature unblinding of subjects' treatment allocation is never appropriate in an ongoing study. Hands for true. Hands for false. And you all have it again. Of course, emergency unblinding is absolutely necessary. So premature unblinding of subjects' treatment allocation may result from which of the following? A, failure to mask dosage and administration variables, mismanagement of IRT access privileges, IMP labeling and shipping documentation, or all of the above. Those takers for A, for B, for C, and for D. We clearly make this too easy on you all. And that's the end of our talk today.